Okay.
1 second. Sure. Oh. Hold on. Okay, can you see the
Yep, perfect,
present
Professor
screen?
Kiyoko. Professor.
Okay.
Yep, perfect.
Okay, so thank you for giving me the opportunity to talk about our new exciting project that started in Japan about the Human Glycome Atlas project. And this is really aiming toward understanding the extended central dogma, which I will describe. And in order to discuss this, I need to give you some background. So I'm from Sobe University in Japan, and I've been really focused on linked open data and the semantic web, where we're trying to integrate life science data on the web to make it easier for bioinformaticians and researchers to understand life sciences. But the life science, this LOD cloud is not only covering the life sciences, it also covers many different domains, including geography, government, linguistics, publications, and social networking. But as you see, these are lots of networks, and they all, especially on the semantic web, they include information that incorporates the meaning, the relationships between these different data points. So it's a really useful tool, especially for heterogeneous data. This is the LOD cloud for just the life sciences, and you'll see that there are lots of, it's a very meshed network of information. But what was interesting was that there was a paper in 2021 that analyzed this life science LOD using a meta-analysis. And they found that there's a few major nodes that really provided lots of integrations with different data sets. So this NBDC is this Japanese funding agency who has, they've been really promoting life science integration, database integration in life sciences. And so I've been fortunate to be involved in this. And so our project, GlyToucan, as you see here, has also been trying to really integrate all this information. But to step back a little bit, so in order to get to the LOD cloud, we have to understand what the data is. So I want to explain what the extended central dogma is. So I think everyone knows the central dogma, which is how DNA are transcribed into RNA, and then RNA is translated into proteins. That's fine. So many high school students already know about this. But it doesn't stop there. So there's protein that are, after they take their three-dimensional form, they are also post-translationally modified. And so there's lots of different types of PTMs, phosphorylation, methylation, acylation, smolylation, ubiquitination, and they all modulate protein function in some way. But the largest type of PTM are glycans, glycosylation. And so these are very diverse molecules that are attached to proteins, and they are very diverse. So this is from a online textbook called, it's not written here, it's The Essentials of Glycobiology on the NCBI bookshelf. But this is only a snapshot of mammalian glycosylation. So we're not even covering bacteria and plants, which are much more diverse. Just even look at the mammalian system on the exterior membrane, cellular membrane, there are transmembrane proteins, and they have lots of different types of glycans on them. So this key indicates the different monosaccharides, which are chained together to form these complex branching-like structures that are on the surface of cells, and they're on every surface of every cell. But they are not attached. They're not just attached to proteins, but also to lipids. So they form glycosphingolipids, proteoglycans, as well as what's called GPI anchors, which links between proteins and lipids. And very interesting. But they're also internal, intracellular glycosylation as well. O-glucnac is that one well-known one. But unlike the central dogma, the extended central dogma needs to consider the biosynthetic pathway and how glycans are formed. So this, in a sense, DNA can be easily copied to RNA. RNA, you can figure out the protein from RNA, but for glycans, it's not so easy because these glycans are synthesized using a large variety of different enzymes. So this is the cytoplasm, the ER lumen, in the ER, the glycans are synthesized one at a time by different enzymes indicated in the red. And depending on the substrate and the donor substrates, they are formed one at a time to form these complex structures. And this is just one type of structure. There are also many other types of N-glycans, or all types of glycans as well. But eventually, so they get attached to the protein, which comes from the ribosome, and then attached, and then this goes out into the Golgi, where there are other enzymes which further process these glycans to form a large variety, the variety I just showed you in the previous slide. So it's very complex, and so that's why it's been very difficult to understand glycobiology, like these glycan structures. But even a long time ago, I think this is like 20 years ago, this kind of picture has been taken of a cellular membrane. So this hairy stuff, this is the cell, and on the outside of the cell, this hair is all glycans. So you can imagine how you can see how all the glycans are just covering the entire cell. So in order to do any kind of intracellular communication, you have to go through these glycans. So you can't ignore them. You can imagine how viruses need to enter the cell to proliferate. So they have to go through these glycans in order to infect cells. So even the coronavirus has many glycans involved in the infection, SARS-CoV-2, of course, and many others, bacteria and antibodies. They all go through and have to interact with these glycans. But up until now, it's been really difficult because of the technologies. There's no sequencing technology for this, and so the Human Glycome Atlas project is aiming to be able to develop that functionality. But stepping back a little bit, we have been working in Japan in my lab on this Glycosmos portal. It's been a joint collaboration across Japan, and now it's been recognized as the official portal of the Japanese Society for Carbohydrate Research. This again, we've been funded by NBDC as well a little bit with the NIH. But what is in Glycosmos is that we have two major sections, actually 4 sections, but here's listing 2 of them: repositories and data resources. So repositories are where users can upload data and supply information and data to get accession numbers. So Gly2can is the repository for glycan structures. So now we have IDs, accession numbers for every glycan. Glycome is a new repository we've just published, or in the process of publishing, for glycoconjugates. So these will assign accession numbers to glycopeptides, glycoproteins, glycolipids, etc. And then the current technology for sequencing glycans is mass spectrometry. But there's NMR as well, but mass spectrometry technology is the most high throughput at this point. So we have repositories to upload glycomics and glycoproteomics data to so that people can get and publish their data, but also get accession numbers. And so GlycoPost and Unicarb DR help to facilitate that. But this is for people who are already studying glycans. For those who want to understand and learn more about glycans and find out their relationships with other information, we have this data resources section. So you can see these icons will indicate what type of data that you're looking at. It's a glycolipid or glycan binding protein, which are called lectins, pathogens, entire glycomes, glycan structures, glycoproteins, and genes, the enzymes I mentioned before. Just a general overview, this is a figure to illustrate the relationships. So we have glycans in the center. Glycans are synthesized by glycogenes, and we have a Drosophila gene database, but also other gene databases. And then genes are of course central dogma. So proteins become proteins, but these proteins are glycosylated themselves. So we have a relationship this way as well as this way. There's glycolipids. And then the interactors. So the glycans are recognized by different lectins or pathogens. And so we have this information available from these resources. And then overall, we have different pathways, reactions, disease information that kind of relate to all of this information. So it's coming on the outside. So Glycosmos, we publish updates every 4 months. The next one for Glycosmos will be out soon in a week or so. But the main interface here allows you to do a cross keyword search across all these different data resources. And if you're looking for a specific glycan, there are different ways to look for a particular glycan, which I will describe it in a little bit. And then the major resources are here. So you can see if you're, depending on what you're looking for, you can just go to these different pages. So this for one example, there's so much data, I don't have time to go through all of them, but this kind of exemplifies all the data that's been integrated into Glycosmos. So we've taken data from Reactome, the Reactome database, which is already RDFI on the semantic web. So we have that information. So if you do a search, a keyword search, for example, for keratin, and then specify the species, then you can get this kind of pathway. Now, this pathway is not the same as what's in the Reactome database, those who know. This is actually automatically generated on the fly. So the network information is in RDF, is in the linked format. But what we do is we take that and just place them on this JavaScript based tool, which has the cellular membrane, the nucleus, and the cytoplasm, and then we just place the network within the appropriate locations. This green area means that this is actually in the lysosome inside the cell. So this is a degradation pathway. So keratin sulfate proteins get internalized into the cell and then degraded in the lysosome, and these are different enzymes that work on it. But you can click on these different nodes in the pathway. So if you click on this, for example, node, then you'll see that this is a glycan. It has a glytucan ID, and this is the structure. Or you can click on the edge, that indicates that this is the enzyme that catalyzes the reaction. So this is a beta-galactosidase, and this icon indicates it is also a glycoprotein. So this is an enzyme that works on glycan, but it itself is already glycosylated. So you can see the actual glycosylation patterns of the glycosylated structures on that. So there's more information, but this is just showing the glycosylation sites and the glycan IDs that have been published in the literature to indicate what kind of glycans. This is monosaccharide compositions. So this is the most detailed we can get from mass spectrometry at this point, but we were able to encapsulate that into the database. The circles indicate proteins. So if you click on this, then you see that the core proteins that are being degraded in the lysosome. So in this case, this is a complex. So we have different types of these keratin sulfate core proteins. This one also indicates it's a lectin. So this has also further links to the lectin pages indicating what kind of glycans that it is known to recognize and bind to. So this is one exemplar of how we can integrate glycan structures with glycogene information and glycoprotein information and lectin information, all from a single resource, which was not really possible 10 years ago. So it's been an exciting path. This is the tools that we have in Glycosmos. So we have a number of tools for drawing, and for analyzing glycan pathways and look at the glycomes of different species, and other tools. So this is the sugar drawer tool, this top one. This is the simplest interface for drawing a glycan. So you just click on these monosaccharides and then attach them to each other in different conformations. If you don't know, you can use the question mark, and then you can search and then get the ID if it already has an ID. You can also export it as a string, and if you have another string, you can import that in the string and draw and see how it looks like. But you can also click on the ID if it's available, and jump to the actual entry page, which has more information. So this is the glycan entry page for this glycan structure, which is very famous. It's called the Cialis X structure, which is also known as a biomarker for many diseases. You see the species that it has been found in, IUPAC string, the mass, and then other content, which I have no time to show now, but we can We have information about what enzymes are known to synthesize or generate this glycan, the core proteins that it is found on. It's an epitope. If it's a lectin, you have data for the lectin frontier database. We have glycome information, so we know which tissue or organs it is found in human or mouse, pathway information, drug information, etc. And of course, literature information. The other drawing tool is called Glycan Builder, which is a little bit more complex. It's more for technically inclined, those who are more familiar with glycosylation. So for example, if you want to input a composition, this is not possible in the sugar drawer tool, but if you want to input a composition, you can do so. And it'll calculate the mass for you, and then you can search for it in Glycosmos or Glytucan, and then clicking on the search button, this will show you the entry page for that glycan. So in this case, there's no other information. We don't know the species or anything. We can't draw the 3D structure and the atomic structure, and there's probably not much information in these other sections. But there's links to PubChem, which has a lot more other information as well. So the GlycoMaple tool is also a tool that we've been developing in collaboration with Dr. Fujita at the Gifu University. So he's a glyco scientist, and he is interested in glycogene expression data. So we have lots of RNA-seq data, of course, in many different public databases. So what you can do is, because we don't have a sequencer, you can use RNA-seq data to estimate the glycome. So basically, it visualizes the enzymes in different pathways. So there's lots of different pathways. These are all manually curated pathways, and it'll display the highly expressed enzymes with the thicker arrows like this. So using this, you can estimate what kind of glycans you would find in that sample from which you extracted the RNA-seq data. So in this way, you can also compare, for example, cancer samples against normal samples and see which enzymes are more highly expressed. Usually the Lewis structures might be more highly expressed, for example. And so this is a nice tool to see across all the different types of glycans possible, the different expression patterns and different glycans that you might expect. This is the Glycome Atlas tool we've developed a long time ago for human, mouse, and zebrafish. So if you click on any of these organs, you'll see the different glycans that have been identified there. And conversely, if you click on a glycan, it'll also display in pink the other organs which also contain that glycan. So this is a nice, fun tool to teach about glycans and glycobiology and show, in a visual way where the glycans are. Of course, these are also linked to Glytucan and Glycosmos, so you can click on the glycan itself to get more further information, the details. This is another tool in collaboration with the AIST, where they have been developing a lectin microarray. So lectins are analyzed. So I don't know if you can see here, but you can see these small locations in these slices of the mouse, of different mouse tissues. If you mouse over or click on any of these small sections, different sections of the same organ will have different lectin profiles, meaning that the different glycans are located in those different sections. So if you homogenize one tissue, you're going to be mixing all those different glycans from all the different regions together, and it might be difficult to see the different glycan patterns that might be found in different areas of even the same tissue. So in this way, you can compare different areas and looking at the lectin binding affinity, lectins are known to bind to certain specific glycans, and so that will give you an idea of what kind of glycans might be found in those specific areas of the tissue. So this is also a very useful tool to analyze glycosylation. So overall, we've been working with PubChem because glycans are very complex molecules, but they have a lot of information about glycans, not just glycans, but genes as well, and publication information. So we are continuously working together to integrate our information and link with each other. So we published this this year. Also, we've been working in what's called the GlySpace Alliance. So this is an alliance between the US, they have what's called the Glygen project, and in Europe, the Glycomics at Ecstasy project. These are both portals funded by their respective areas to try to integrate glycan-related data. Of course, so one location can't cover everything. So we are all agreed to share our data with each other, and also to make sure that everything is trustable, that the data is trustworthy, and making it all freely available. So this we've tried to really work together internationally to provide a solid foundation for glycosciences. So finally, I'm going to talk about the Human Glycome Atlas project. So this is a project that has just started in April this year in Japan. So it's actually the first time that a life science-based project was funded under what's called the Large Scale Academic Frontier Promotion Project, or the Next Frontier Project. Up until now, this Next Frontier Project has funded physics or astronomy, very physical types of projects, and life sciences has not been considered. So this is a very exciting time. This is the PI. I am not the PI. This is the PI of this project, Nagoya University, Dr. Kadomatsu. But it's a Japan-wide project. Just to cover the history of different projects. So as you know, the Human Genome Project was started back in the 1990s. And finally, we got we could understand the whole human genome by the 2000s. And now we have a reference human genome, right? And several different reference genomes for human. And now that's really contributed a lot to genomes medicine and precision medicine and customized medicine. There was the Protein 3000 project in the early 2000s, and now there's a Human Protein Atlas, which has lots of information about human proteins and from different perspectives, single cell, tissue level, etc. And during this time, ABO blood type, which is where glycans are very important, has been studied, and Japan has for a long time covered glycochemistry and glycoscience research since the 1990s. And then with the Glyphosate Alliance forming in 2018, it was about time to start the Human Glycome Atlas project. So this project was selected as a roadmap project in 2020 by the Japanese government. And we finally started the official project this year, 2023. This is a 10-year-long project, and it's really exciting to be able to start working on a reference human glycome. We're hoping to aim for by the end of this project. This project is also the foundation of it is the J-GlycoNet, or Joint J-GlycoNet Research Network, collaborative network in Japan, which I will also describe later. So, okay. As an overview, so there are 3 objectives for this Human Glycome project, Atlas project. The first objective is to create the glycan information infrastructure. So we want the ultimate aim is to create this knowledge base, which we're calling TOSA. So we're calling it as different segments. So this is the final segment among 4 segments to store all the data that have been generated from segments 1, 2, and 3. Which I will describe in a bit. There's also objective 2 is to create the infrastructure for objective 1. So there's advanced systems that are necessary to construct for TOSA and to establish global standard technologies. And so this is also being worked on. And then objective 3, the ultimate aim is to be able to provide a place where not just the researchers in Japan, but also across the world can come and collaborate and work with us and each other to study the glycome and glycoscience related research. So really aiming for collaborative research in the end. So to go more into detail in subjective 1, the aiming, to construct the TOSA knowledge base. There are, like I said, 4 segments. Segment 1 is aiming to develop a precise human glycoproteome map. So we're trying to map the entire human glycoproteome to understand, to be able to, just a reference glycoproteome for across all human samples. So this is being done by using mass spectrometry and liquid chromatography and generating this map, and then also creating a glycome catalog. So this is just glycoproteomes, but there's also glycolipids, glycosylglycans, free glycans, etc. So the total glycomics will also be performed to get a quantified information about all the different types of glycans in these different samples. And then we have a large cohort, which we're collaborating with different hospitals in Japan. So based on this glycoproteome and total glycome, we will compare healthy versus disease patients to be able to catalog the changes in glycan data across different states of human. So this will be integrated together. And then segment 3, which is still kind of small, it's still started, but we're also looking at the glycoenzymes and trying to perform a comprehensive analysis of those to understand their localization inside the Golgi and inside how they are, synthesizing glycans, and also recording their activity levels across different samples. So all this information will be stored in this knowledge base TOSA by the final year. But the research plan is to across these 10 years to focus first on blood samples. So dementia and aged patients as samples will be collected and analyzed for their glycomics and glycoproteomics data. There'll be automation and robotics used. Of course, we have a 20,000 cohort, so it's quite big. So it will take a long time to analyze. So we need to automate everything. There's also research going on visualizing the biosynthetic mechanisms of glycans. So we can visualize within each cell where each glycosyltransferase or each enzyme is located. So we're going to collect that information as well. And so by the year 5, we will have a knowledge base of this basic information. And so by year 5, we will have an access-controlled database of all this information. And so research collaborations can be made based on that. And then further, other tissues and organs will be covered with other diseases across the latter half to acquire comprehensive data on the human glycome, and further, adding to the TOSA knowledge base, and really aiming towards cell therapy to try to engineer neo cells by selecting specific enzymes, for example, to synthesize specific types of glycans. And tools to support that will also be developed. So we aim to have a open version, open data by the 8th year, which I think is 2030. Well, long time, to be able to support and provide all this information to the open data space. So the ultimate scientific goal is to establish a foundation for the extended central dogma. So, the central dogma itself is not enough to explain life. We cannot, it's not right now. We haven't been able to solve a lot of major diseases based on just the central dogma. Of course, there's RNA and non-coding genes and etc. also involved, but this is also involving glycans. So, really emphasizing that we need to understand glycosylation as well to extend the central dogma to truly understand life activity. And so the 4 pillars of those segments really to establish this glycan information resource will help to support that. So how can everyone be involved? So this project, like I said, will be creating this TOSA knowledge base and the open data. So Glycosmos is based on open science. So any data that can be made open will be shared with Glycosmos, which will then eventually be shared with the GlySpace Alliance. We already have global support for HGA through Academia Sinica, MOUs with Kinoshita, Australian universities. The Human Glycome Project is being promoted by Gordon Lauk and Rick Cummings, as well as the University of Lille. So really, not just establishing a extended central dogma, to promote interdisciplinary research, we want to create a new life science community, not just domestic, but also globally, to promote this extended central dogma. And in order to do that, one of the first steps is JGlycoNet. So like I said, JGlycoNet is a collaborative network, science is spelled wrong here, cooperative network across Japan, focusing or being supported by IG Core, the XL's Research Center, and my institute, the Glycan and Life Sciences Integration Center at Soka University. So these three institutes are working together to do joint research. So actually right now we have a joint research call. So people here who are interested can apply. The information is at this URL. Oops, sorry. At this URL for any joint research. Due date is January 31st, but the exploratory type is available for domestic and international joint research on a wide range of themes involving glycans, promoting research across all life science fields. So it doesn't, you don't have to be a glycan person. You only need to collaborate with someone in JGlycoNet, find a host researcher in the JGlycoNet, and you can get funding for up to 1 year to study where your research can link with glycans. So of course I'm a member of the JGlycoNet, but there are also many other members in JGlycoNet. So you can go to the one-stop consultation desk, or you can ask me, it's fine. But this consultation desk here is a contact point for any researchers who want consultation on glycan-related research. So you can fill in this form, and everything will be strictly confidential, and it's very confidential research. So, but this consultation desk will help to assist in connecting you with JGlycoNet researchers and collaborative fellows to help team building and foster collaborative research. So these are two resources which we are providing, and which we hope will allow many people to help participate and take advantage of this Human Glycome Atlas project. So there's a little bit of time left. I want to just quickly mention that we're not only working on a human, myself, the Human Glycome, of course, HGA is of course human, but I've been also interested in the microbial glycans as well. So we've been developing this database called MicroGlycoDB. It's in the alpha stage right now, but we're working on the publication for releasing this database. It's also linked with Glycosmos. Basically, it's a database of glycan-related information in microorganisms using semantic web. So we've had collaborations with researchers that have been studying all these different organisms. So for example, Bifidobacterium bifidum, we've received their data like this as images, and then you can click on these different images and see the different genes that are involved in generating those glycans. So this is very specifically for this species. You can see the type of the pathways that they have discovered in generating the glycans in this species. By clicking on the glycan list, you can see the list of glycans that have been identified in this species. There's the detailed entry page, which also has other links to other information in other databases, as well as for the details about the genes themselves. So this gene NAGB facilitates this reaction, deaminazing this glucosamine 6-phosphate into this fructose 6-phosphate, for example. So going on, we also have information about Bifidobacterium longum, which degrades larch and gum Arabic structures. So these are the different enzymes involved in this species, and you'll see the list of genes and glycans there. We also worked with Christine Zemansky, who is a Campylobacter jejuni researcher. So if you click on either the cell body or the flagellin in the Campylobacter image, then you'll see the different glycan types that are known to be involved or found in those locations. You can also then click on one of those types of glycans, and it'll display the pathway to synthesize those glycans. And so these are all, curated information checked by experts, and you have the whole list, which is also downloadable. For Cryptococcus neoformans, we have not so many glycan information, but we have the gene information, and so we display this. For Mycobacterium abscessus, similar to Campylobacter, we have the images where you can click and get the pathway information, and same thing for Mycobacterium tuberculosis. So this was a very involved project. We had to work really closely with microbiologists to get their information and process the information and make it clean and consistent to put it into the database, and that was very difficult. So we've been also working on a way to make that easier. So we've been working on what's called the microglyco curator system. So this is also an alpha version. It's still very, very early stages, but we're hoping to create a way for researchers to easily curate their data to be able to put it into MicroglycoDB. So after logging in, you can register your species, even up to the strain level, and then there's a template file which can be uploaded and submitted, and it'll be displayed here. There's some IDs that can be automatically filled in using our template. And then based on that information, you can automatically get the images. So in this case, the information that we received or has been uploaded was about genes in the cell outer membrane. So the cell outer membrane has been highlighted in light blue here. It's hard to see. But so we're able to do this kind of automatically. We don't have to draw those images ourselves. They're already provided by the SIB library. And we can also generate the pathways automatically from those the templates. So we can just draw this. I think this using Cytoscape to display the pathway. So we're working on that and hoping to promote that as a way for more microbiologists who have glycan data to add that information as well, and that'll eventually get into Glycosmos. So in summary, I guess it's okay, I guess time-wise. The HGA project will produce the first ever reference human gene glycome, I hope. So that's the goal, to be able to provide that by the 5th year. By linking the HGA data from the TOSA knowledge base with Glycosmos and linked open data, we will be able to be providing both informaticians and researchers in general with a large data set encompassing the extended central dogma. So, and it'll be important, especially for researchers, to develop user interfaces that make it easy to access the information that they're looking for. So it's just so much information. And of course, all this data is very exciting for AI and generative models to extract new knowledge for understanding life. There's lots of hype, and I guess we'll also have lots of sessions later in this conference to understand how we can take advantage of AI, and we're hoping to provide the data to allow AI to work and get information that is meaningful for the life sciences. And like I showed with the MicroglycoDB, we're not just focused on human, but also environmental engineering, environmental sciences. I didn't talk about plants, but material sciences, oceanography. There's so many places where glycans are involved, but the data has not been organized in any database. So we're hoping that this kind of technology in Glycosmos and Microglyco Curator will help to facilitate more tools to help organize and supply that information that is needed for knowledge production. So right now we're looking on single cells. We're looking at cells at the cellular level, but this can be analogized, to the entire world where there's so many different things involved, and we're hoping to link all this information using the LOD technologies. So finally, acknowledgements. The MicroGlycoDB collaborators are listed here: the University of Georgia and University of Lille, and in Washington University in the US, and Kagoshima University and Kyoto University in Japan. They've been really helpful and generous to provide their data. And the Glycosmos project has been supported by the NBDC, JST, and the Japanese Society for Carbohydrate Research. And these are the current people working on the project, but we have many more who have worked in the past. It's been a long project and very fruitful. And just as a plug for next year, the next year's INCOB, JSVI, GRW, APBC, ISCB Asia will be joined together in the first ever Asia and Pacific Bioinformatics Joint Conference. So as one of the organizers, I welcome you to check out this webpage. We'll have a call for abstracts early next year, but please keep the dates available so that we can see you in Okinawa, very beautiful Okinawa, and have an exciting time to create synergy across the Asia and Pacific regions in bioinformatics. And we do plan on having a glycoinformatics session there. So it should be exciting, and I hope to be able to promote and explain more about progress in glycoinformatics and human glycome project. Okay, thank you. And that's it. I'll take any questions.
Okay, thank you very much, Prof. Kiyoko. I don't know about how others are feeling right now, but I feel absolutely overwhelmed with the amount of work that has gone into this. No, this is amazing. This is very inspiring, and I really love this because I think it's a very, very good reminder that we don't all work in solos, right? We work together with everything. Like I was absolutely astounded. So I really love the talk. So that was excellent. So I think while others are if you have any questions, just a reminder, if you have any questions, please feel free to put in the chat. Meanwhile, I think I'll kickstart off first. Prof. Kiyoko, I really love the talk, and I'm not an expert in glycan biology, but I just want to ask a few general questions, right? Because just now in the Glycom Atlas version 5, you mentioned that you can actually see different glycosylation patterns depending on which kind of organ. So just asking out of curiosity, like, is it the case that different organs will have different glycosylation patterns, and is there any implications to diseases? Yeah.
Oh yes, definitely. Even the same cell will have different glycans at different times. So it's really, glycans really indicate the status of that cell. If it's healthy, if it needs energy, it's very dynamic. And so we're hoping to be able to that's why it's a very difficult project, but we need 20,000 samples in order to get this entire reference genome, to be able to cover all the different stages that could be possible for every organ. So yeah, that's like I said, in the lectin microarray, you saw even in the same tissue in different locations, you'll have different glycosylation patterns. So definitely different cell types, different organs will have different glycosylation patterns. But, yeah.
Yep. And I think just now, just something that I thought was quite cool, because I actually work on chemoinformatics, and I think something that reminds me was the sugar drawer. I really love the concept of sugar drawer. That was beautiful. It's an absolutely beautiful interface. Yeah.
Thank you.
Yeah. Just us. Yeah. So hang on. There's one question from the floor. So Harpreet is asking, there's a call for research projects in 2024. Can the masters join for their first few months on this research project? I think just now there's also a few people who actually personally messaged me to ask about whether or not we can share, like, a bit more about your research, about your call for research project. Do you mind to share a bit more on that?
Oh, definitely. So yes, the collaboration grant here, unfortunately, you have to be a faculty, have a position at an institute to apply. But if you have students, you can add them to your research group, and so apply to. Yeah, you can have students come, and of course you can join. So you can add your students, and then figure out how we can have your students come to Japan and collaborate with us. So definitely, and please send out the word that this call is out. That'd be great.
Great. Okay. Any other questions from the floor? So from Aini, how can glycan in pathogen contribute to their pathogenicity? What is the role of glycans in pathogenic bacteria?
So bacteria are very smart. So they can mimic the glycans in the host. So that's how they evade, being attacked inside the host cell. So there's lots of, I'm not a pure microbiologist, so I only know from what I hear, but they use glycans and if they're infective, to pathogenic, they mimic the glycans in the host. Actually, they even use the host's glycosylation machinery to generate the glycans in the host so that they're not detected.
Prof. Kiyoko, I think, yeah. So there's a comment from Akriti. So thank you, Prof. Kiyoko, for taking us through your work. It has been comprehensive and beautiful presentation. I absolutely agree. It's such a beautiful slide. As a PhD student, I wish to ask your work, is it bound by one umbrella research, biological, objective and diverse fields of computational biology? So shall we approach for our research proposals and objectives similarly?
Is this regarding the J-glyconet? Maybe.
I guess what. So Akriti is one of our student ambassadors from AP Bionet.
Yeah, she's mine.
Okay. I guess she's probably asking.
Research proposals. So she talk about J-glyconet, but overall work. So yeah, no, my research, I like to go everywhere. I'm kind of scatterbrained, so I'm interested in many different things, but always focused on glycobiology in the end because it's connected to everything naturally. So but it depends on the person. I don't think there's one answer for everyone. If you're interested, in one specific thing, I would encourage you to focus on that and go forward at that. And naturally, if there's others, researchers that are experts in other fields but kind of related, then you can have collaborations with them. You don't have to be an expert in all of that. I think it's important to have diversity even in project teams to be able to have really exciting research.
Yeah, I actually I really do like what you said about Kiyoko, right? It's not just about getting stuck on one single field, but being able to have different knowledge in different fields and also having diverse skill sets as well, right? It's not just about biology, but it's also about combining. I guess there's a lot of coding behind whatever that you're doing as well, right? So having all these diverse skill sets, I think that's very, very important. I really like what you're doing. I initially when I was looking at presentation, I thought that it's just about humans, but suddenly you veered into microbes, and I was like, oh my god, there's so much work. But I think that's amazing. Okay, any other questions from the floor? Okay, calling once, calling twice, calling thrice. Nope. Okay. All right. Just to let everyone know, because I've been receiving a lot of spams on personal messaging asking me to share a Prof. Kiyoko slide. Just FYI, we will probably share the recording with everyone. Okay. So I think we will do that in due time. Okay. If there's no other further questions, then Prof. Kiyoko, I just want to say thank you very much for that presentation. That was absolutely mind-blowing and very inspiring. So thank you very much. Well done. Thank you. Okay. All right.